everyone, and welcome to today's Seed World Innovation webinar series. My name is Alex Martin, and I serve as an associate editor for Seed World. And today, I'm happy to be your host. Today's theme is putting the power of prediction in the hands of plant breeders. Before we begin, I'd like to thank Benson Hill Biosystems for partnering with us on today's webinar. Presenting the webinar today is Dr. Kendra Meade. Kendra is the Director of Customer Success at Benson Hill and works very closely with partners to help them get up and running on the company's predictive breeding platform. Kendra has a PhD in plant breeding from Iowa State University and 7 years of experience in predictive breeding. She has worked on implementing predictive breeding programs in over 15 crops, including polyploids and clonally propagated crops in addition to row crops. During the presentation, you'll likely have some questions for our speaker. Please type these into the chat box at any time during the webinar, and we'll address them during our Q&A session we'll hold after Kendra finishes her presentation. Also, during the presentation, we'll have a few poll questions for you. They will pop up in your chat box for you to answer. Today's webinar is being recorded and will be made available at seedworld.com following the proceedings. In today's webinar, you'll learn the differences between predictive and conventional breeding, the specific steps to genomic selection, and a strategy to apply predictive breeding to your breeding strategy. We'll also be tackling some predictive breeding frequently asked questions. We have quite a full schedule, so go ahead and take it away, Kendra.
Thanks, Alex. And thanks to all of you for joining us today. We're really excited to talk with you about the possibilities for predictive analytics and predictive breeding within your program. To get started, I just want to frame this up so that you kind of know where I'm coming from and how I think about plant breeding and how we at Benson Hill think about plant breeding. And when we approach this, we're really looking at it in terms of operations and organizations are often 90% of your battle when you look at a plant breeding program. So when you approach it from that standpoint, you approach it, of course, with the first question to ask is, what's your goal? Do you want to have an improved taste on your crop? Are you looking for increased yield? That could be the kind of yield we talk about when we think about row crops. It could be the kind of yield when we think about more like a vegetable crop, so marketable yield. What kind of unique quality characteristics are you looking for? Do you have some enhanced nutritional aspects in mind? And what kind of enhanced performance do you need under varying growing conditions? And those varying growing conditions could be anything from the kind of differences in environments that we see on a year-to-year basis in a standard field crop to the very specific things associated with greenhouses and the, say, light conditions in a greenhouse atmosphere. So when we do this and we figure out what our objectives are, we can then identify our constraints. And our constraints are really around things like time, man hours, budget, all of those things. And so anytime you implement a new technology, you of course have to make sure that that new technology is going to fit within your objectives. So it's going to enhance your role or your goals and your positions towards your objectives. And then it's going to help you meet those objectives within your constraints. So when I go through this talk, we're going to talk about planning your breeding program around your objectives and the limitations of your plant system and how predictive analytics can help you do that. So that, of course, brings into call the idea of the plant breeding cycle. So when we start out with plant breeding and we start to think of traditional plant breeding, we're thinking in terms of a phenotype. And of course, if you break that phenotype down, particularly when you think of it from a statistical or a modeling standpoint, we're looking at having a genotype and a genotype by environment interaction and the environment. So as we go through this cycle, we collect all of this important information so that we can make it through this cycle as quickly as possible and with as much accuracy in our selections as possible. So we're collecting information around phenotypes, genotypes, and environment. And the difference between a traditional breeding program and a genomic selection breeding program really might come into play around collecting information around those genotypes. So where you might be running a few markers for, say, a marker-assisted selection program, or even no markers for a traditional program. Now we're going to talk with you about having to actually genotyping the entire genome. And then we're going to use all of that data that we're collecting, those phenotypes that you're collecting and those genotypes, and we're going to combine them together to do some pattern identification. And that pattern identification is really where we're getting into some predictive analytics, because once you identify your patterns, you can actually make predictions using only a subset of the information for other lines, in this case, the genotypes, and you can make those predictions out onto how you expect those lines to perform. So if you have some genotypic information on a subset of line or on all of your materials, but then you only have your phenotypes on a subset of those lines, you can use all of this together to actually get a feel for how are those lines that you've only genotyped going to perform overall. So that kind of gets into this idea of what is genomic selection. And so genomic selection is essentially a breeding technique that uses predictive analytics to improve your speed and efficiency. And the great thing about genomic selection is that it actually fits within your breeding program. So all it is creative ways to apply predictive analytics at each phase of your breeding program. So you start out with making crosses. You make crosses that we've predicted to produce diverse, high-performing progeny. We evaluate those progeny so you can reduce your trialing costs by only trialing a training set, or by using the previous generation's trialing results to be able to actually make selections in an off season where you might not have been able to do that before. You can then advance progeny, you can enter commercial trials faster. All of these things are made possible by predictive analytics. So for those of you who aren't all that familiar with genomic selection, or for whom it's basically been something that you've heard somebody mention at some point in time, and like, maybe I should consider starting that up in my breeding program, just giving you this slide just gives you a feel for how common it's become. And what we did here was we went out, we did a Google search for publications with genomic selection in the title. And you can see where 14, 15 years ago, genomic selection wasn't well established in the literature. These days, though, it's highly established. It's a very common thing. And you can find a lot of literature out there to provide you with guidance on how to go about refining your genomic selection techniques. And we here at Benson Hill have built tools out around the idea and around some of this literature to help you make predictive analytics and genomic selection possible within your breeding program. And so that brings us to the idea of how does a breeding program with genomic selection differ, and how is it similar to a traditional breeding program? And as I mentioned before, the whole point of genomic selection is that it's going to enhance and augment what you're already doing. And the way it's going to do that is by having this training set. And this training set is essentially a pool of data that includes the genotypes and phenotypes of a lot of your materials. And you can use that training set to model and run genomic selection onto lines that you've only genotyped or that are very early in your program. So maybe you don't have a lot of great data around them. This is going to differ from what you're doing perhaps now, or if maybe not, if you're already running genomic selection. But for a traditional breeding program, your crosses and your cultivar development are largely dictated by your phenotype data and by your breeder's knowledge. And then once you get through those selections, you then trial for multiple seasons to make selections. Whereas with genomic selection, in general, we're going to be able to focus in on our high potential candidates because we're using a lot more information around the genotypes and phenotypes. And we can decrease our trial plot numbers and seasons because we have this training set that's already informing our decisions. We can actually increase our selection pressure in even in the off season sometimes to allow us to speed this process along. So that brings us to how do you perform genomic selection and how do you make it work within your breeding program? So there's some key steps to genomic selection. And I do just want to point out, we've got some fun survey questions and we are going to share those results with you. So if you could tell us if you're working in a self-clonal or a hybrid crop, that would be awesome. Just know that, we're really excited to work with you and talk with you about the possibilities of any of these different types of crops. But some of the key steps in genomic selection include collecting data and making it available, understanding what you have. So once you've got all that historic data pooled together, you've got it organized, which, by the way, is a big task and one of the hardest things that you'll do as you tackle the idea of genomic selection. And we're happy to discuss with you the best ways to do that. You can begin to understand what you have. Once you understand what you have, you can then use that information to create models, make predictions, advance, make new crosses, and eventually enter your next cycle of genomic selection. So that brings us to step one, which is collect data and make it available. And there are two different avenues that you're going to collect data on. So you're going to collect data on your genotypes, and that's your top arrow here. And we recommend often that you begin by sequencing your founder lines. So if you don't already have genotypes on a lot of your materials, or maybe if the genotypes on your founder lines, and by founder line, I mean a line from which you've drawn a lot of your parents and progeny within your breeding program, it's kind of going to be the base of your breeding program. So if you go back through all of your nursery books, you're going to find this line again and again and again in your pedigrees. We sequence those founder lines, and then you can use that sequence data to identify marker sets for progeny genotyping. And the nice thing about using the sequences of your founder lines to identify your marker set is that your marker set becomes very specific for your breeding program. So you can really design that assay and direct all of those genotyping dollars towards markers that you know are going to be informative for your breeding program. Once you get past that, you genotype your progeny, and then you onboard your genotype data into a tool that makes it easy for you to use and view the kind of genetic diversity, and then eventually use that data in models for genomic selection. At the same time, at the same time or even before, you're going to be phenotyping your founder lines and phenotyping your progeny. And this step, in my mind, also includes that organization of that historic data. And with the breeding programs I've worked with, this is a big task, but it's a really important task because that historic data that you've been sitting on is one of your most valuable assets. It tells you a lot about where you've been and where you're going. It tells you a lot about how your goals have changed over the years, and it can be the basis for your analytics. So the sooner you can get into that historic data and start understanding what you have, the better. And so once you do that, and once you organize all of those phenotypes, then you can then onboard that into the same tool that you're using for your genotypes, and then merge them together and start running start seeing the results of genomic selection. The next step in this process is to understand a little bit about what you have. So as we look at this slide, we've got two different parts to this slide. We have the genetic diversity of the germplasm. And so this is just a principal component analysis. It's the plot off of the first 3 PCAs from a principal component analysis. In this case, this is a PCA of the genetic distance matrix, which means that each dot on this particular plot indicates a line. And in general, the closer 2 points on this plot are, so 2 lines are on this plot, the more related they are from a genetic standpoint. The nice thing about these plots is that often you can start to identify patterns within them that you can relate right back to your breeding program. In this case, this is actually a plot of some diverse soybean lines from the USDA. And what I found was that as I looked at the country of origin for these materials, they all cluster very nicely together. So the lines that are in green here are actually all the lines that originated from Japan. If I had, for example, highlighted all the lines from India, I would have found them falling over here in this cluster. So this translates to your breeding program. For example, it could be something like you might find that your maturity the materials from your different maturities all cluster together. The materials from your different market segments might cluster together. And the reason for that is because they're originating from the same founder lines. And so you're going to find that you can see a lot of patterns about your history of your breeding program and the relationships among the materials that you're working with by looking at this genetic diversity. At the same time, you want to look at the phenotyped and genotyped lines. And so what we've got here is a table from Breed, the Benson Hill tool for genomic selection. And we have a table of lines. Some of them have phenotypes, some of them don't. So the lines with phenotypes are the ones that have actually been trialed out in the field. And what I see here is a set of analyzed phenotypes. So we've taken the raw data, analyzed them, accounted for blocking effects, accounted for environmental effects, and now we're seeing the end result of this. And we have alongside them the lines that are only genotyped. So those lines haven't been trialed out in the field, but now we're going to take the information from those genotypes and we're going to actually use those genotypes as well as our phenotyped data to make predictions. So the phenotype lines become what's called our training set, and the genotype lines become our target set. So now that I've looked at my genetic diversity and I've looked at my phenotypes, and by looking at my phenotypes, I mean I've looked at the least square means values or the BLUPS values on those. I've looked at the variation within my materials and how those different variations relate to each other. Now I want to go identify my training set and my target set. So I start out with my target set. My target set in this case is a set of materials for which I have genotypes, and I have either no phenotypes or very limited phenotypes. And I want to predict their breeding value into coming environments and into coming years. So I have this target set. I've identified it in my principal component analysis. This is, just to note, this is actually the same PCA plot that we were looking at before. I just rotated it in the tool so that you can see the training set come to the front and the target set come to the front. And so now that I know where my target set is relative to my other materials, I can look for a training set or a set of lines that has really good phenotype data on it as well as genotypes, and I can determine where they fall. And so once I identify that training set, it should be around 200 lines in general is a good training set size. Then I can move on to the next step in modeling. So the next step in modeling is to create a model, and I'm going to train the models and compare performance with cross-validation. So I've got my training set, and now I need to identify which algorithm I want to use. And here we've got a series of algorithms that I can choose from. I've run 3 different algorithms all on the same training set so I can compare their performance. I'll let you in on a secret for genomic selection though. In general, particularly for smaller breeding programs, it's really more important the data that you put into the model than it is necessarily the algorithm. And if you can see the numbers on the slides here, you can see that the numbers that are given from the cross-validation scores in general are pretty close for all 3 models. And that's just indicative of that, of how important that data is relative to how important the algorithm is. But if we look back at the algorithm and we can compare it using a cross-validation. And a cross-validation is really a pretty simple concept where you're going to take your training sets and you're going to split it into 2 portions. And you're going to use one portion to train and predict onto the other portion. So now you've got both observed and predicted phenotypes for each of those. And then you plot those out. You do that about 100 times. You plot them out. You identify the correlation between the observed and predicted values. And that is essentially your cross-validation score. A higher correlation indicates a better cross-validation, which indicates a better overall performing training set. And that's why I say when we look at these, we can see that for each trait, even though the models and the algorithms and training set work slightly differently for different traits, we see that if you look at the same trait across the 3 different algorithms, they're about the same. So really, I could choose any of these 3 different algorithms and move forward. In this case, though, I'm going to go with the random forest algorithm. So that brings me to my next step. And the next step is to make predictions. So now that I have a training set, I have my model. So I built my model off of my training set and the algorithm that I've chosen. I can then move on to making predictions onto my target set. And all that's going to be doing is taking that model, so taking those different patterns that I've identified within that model, and I'm going to be applying them onto the genotypes that are part of my target set. Once I've done that, then I have a completed table that I can interact with. I can sort, I can pull different values out of this table, and I can make selections out of this table because now I have data and a predicted value for all the lines in my training and target set. The nice thing about this is those predictions, they stay in the same units that your actual phenotypes are in. So you can see here, you can still look at flowering date, you can still look at yield in the same values that you were looking at within the same ranges that you were considering before. And you can do sorts and searches. So you can say you're only interested in advancing sets of materials that are greater than 800 units flowering. Those kinds of things are possible as you look at these tables. And so the point of this is that when you make predictions, you can make predictions using the same general decision-making criteria for predictions that you were for the phenotypic values that you were looking at before. Which takes us to this step, which is to identify your advancements and enter your advanced trials. So now you're going to ask the same questions that you asked before whenever you were entering and thinking about entering advanced trials. So those questions include things like, which lines are predicted to outperform others? What lines can I throw away before spending money on trialing? So this is a big one for genomic selection, because rather than spending money on plots, you're actually going to spend a little less money. You're going to spend less money on genotyping. So you're going to have a pretty decent, you can expect to have a pretty decent savings on plot numbers overall. So those kinds of things, you can ask yourself those kinds of questions. As we look at this, one thing that often comes up for breeders is the idea of a selection index. And this is just one point within where predictive analytics might differ a little bit from the way you're breeding right now, where with prediction, you really want to look at the phenotype itself whenever possible. So if at all possible, if you can collect that data and you can analyze that data and then look at the predictions, even if you then pull those predictions out and calculate a selection index off of them, that's okay. But if you try to analyze index selections as a value in general in predictions, that's not going to work so well because you have so many errors that kind of go into those calculations and it can cause some problems. So when we look at all of this, I highly recommend that if you have an index selection, go ahead, go through, run the models, calculate out your values, look at the predicted values before you calculate an index selection, and then calculate that out if you want to. Once you've got your advancements, it's time to make your new crosses. And as we mentioned before, one of the great things about genomic selection is often you can get to this step well ahead of where you could before, because you've got those genotypes and phenotypes on your lines. Let's say you're used to making new crosses at an F6. You might now be able to make new crosses at an F4 generation because you've got all that additional information. So once you have an idea of how those crosses are predicted and which crosses, and you want to know which crosses are going to be predicted to have high performing progeny, you can ask the question within breed. So we have a series of different lines that have been advanced based upon our predictions. And we're going to take those 2 lines, we're going to take the genetics of those 2 lines and their predicted and observed values. And we look at the genetic distance first. So a higher genetic distance in general gives a pretty good indication that there's going to be a wider spread to the distribution in general. We also then want to take a look at what's our mid-parent value. So what's the average predicted value, or what's the average observed value between the two parents? And then we can look at the phenotypic difference between the two parents. This is where it kind of gets a little more interesting, because now we're actually going to go out and we're going to simulate approximately 200 progeny and create a distribution from that progeny. And then once we have that distribution, we can do things like predict out the transgressive segregation ratio. So that's the ratio of lines that you expect to fall into the tails of the distribution. It's a measure of the spread of the distribution and a measure of the indication that you can expect some lines to significantly outperform your better performing parent. You can also look at the predicted progeny standard deviation. So when we consider that, we consider how wide is the spread of this particular distribution. We also look at the population usefulness. I really like the population usefulness score because it gives you an indication both of the mean and the standard deviation of this distribution, which is going to be really important as you think about with your transgressive segregation ratio and your genetic distance. It's really easy to get a pretty high genetic distance and a high transgressive segregation ratio between two poorly performing parents, whereas the population usefulness score, it takes into account the mean and the standard deviation. So you can have a cross and predict out a cross and identify that cross then that has a high mean and a high standard deviation. So you can expect to have a higher transgressive segregation ratio as well as better performing progeny overall. And then finally, we can look at the progeny mean top 10%. And that's just if you did a truncation selection, you selected out the top 10% of the lines and pushed them forward. What's the mean of those predicted lines? So all in all, when we look at this, we can run as many as 10,000 crosses at once within the tool, and we can compare and contrast the different crosses and identify the lines and the crosses that you think are going to be the best parents. Make selections and then prioritize your crosses around that. So that brings us back around to that loop. The next step is you're going to enter your second cycle, or maybe it's just your next cycle of genomic selection. And I think it's really important to do a couple of things once you finish up a cycle. The first thing to do is assess your results. And one of the most important aspects of assessing your results is determining whether or not all of your phenotypes were useful. And that's why I mentioned things like index selection. Breeder score is another excellent example of this. Breeder score is very useful for breeders because it combines so much information for that breeder. The unfortunate truth of it is that that information doesn't really translate that well to an analysis. And so when you think about that and you think about what phenotypes are going to be most useful for genomic selection, you might consider those kinds of things and look at the performance of those phenotypes at the end of your season. You also want to consider your overall cost and timing of implementation. So what went well, what didn't go well, where can you trim some cost, where maybe you want a tissue sample earlier, maybe you want to run genomic selection earlier in your program. And that brings us to continuously enhance. So if you've got the data from last year that goes into your training set, so you're going to feed that right in. So that's part of determining if all your phenotypes were useful. Then you can ask yourself, could you predict earlier or in more generations? And so that kind of gets back to this idea of overall cost and timing of implementations. And then finally, are you ready to expand your use of genomic selection? So can you consider, so if you're predicting earlier or in more generations, can you also consider pushing genomic selection into other aspects of your program, including maybe different market segments, into different breeders programs, depending upon the breadth of your overall organization? All of these kinds of things are really important questions to ask at the end of a cycle. And so this brings me to some key frequently asked questions. And just to give you a heads up, we recognize as we put this presentation together that there's probably going to be a pretty diverse audience. And so we set up the first part of this to be pretty high level. And now I'm going to go a little more into the weeds in terms of some details and some more detailed questions that we're frequently asked as we get moving with customers. If you really want to go into a lot of detail and have a discussion about, say, the details of how to design a training set, and have a deep discussion about that, please contact us. I'd love to have a more esoteric conversation with you around those particular things. But for now, we're going to cover some more information about genotyping assays, when you should run genomic selection, designing training sets, and how to determine if genomic selection is both cost and time effective for you. So first, what genotyping assay should I use? Oh, and just a quick heads up, we got survey 4 question here. So just wondering if you're collecting any genomic data at this point in time. So thanks for your answers. But some different options that we often talk with customers about. The first option is sequencing. For those of you who are familiar with it, and for those of you who aren't for that matter, sequencing is a great tool. It provides you with the most information you can get out of about the genotype of a particular line. So it is going to be highly informative. When you compare the sequences of different lines, you can often identify greater than 100,000 markers. So we often recommend this for founder lines. The reason we recommend it for founder lines is because you can use this as a baseline for that TGBS or targeted array that we talked about, that I mentioned earlier. We also recommend it does tend to be a little bit more costly, but of course then you bring into the idea that because it's so much more informative, it's really great for more than genomic selection. So you can use this for GWAS, other kinds of studies. You can consider putting together some sort of pan-genome if that's something you're interested in. It also has, just from more of a business standpoint, it has the great benefit of there is nothing that is going to fingerprint your founder lines and your breeding program more thoroughly than a sequence. So that provides you with a great deal of protection for your breeding program in a day and age when that's really a necessity. So once we move on from sequencing, we can talk about targeted GBS. So targeted GBS is great in that you can really design the marker number and the assay to fit within your budget, and it's customized to your breeding program. So rather than have a set of markers that are designed more for a diverse germplasm pool, now you're looking at a set of markers that you can expect to segregate within your populations. You can expect them to be informative for your populations, and we can design them so that they span the entire genome. So it's great because it has a lot of information that you can then use as you consider genomic selection, even mapping experiments and things like that as you move forward in your breeding program. So it's a great option. Arrays also are a great option where you have typically around 3,000 to 50,000 markers. Of course, there's some arrays out there that are much, much bigger. I think there's a maize array that's upwards of 600,000 markers. And it's really nice because a lot of these arrays are used for both public and private projects. So you have the added benefit of being able to much more easily combine your data with public data sets if that's what you want to do. It's also nice because many of these arrays are designed for a really diverse germplasm pool, which means that when you have that diverse germplasm pool, if you're frequently pulling in public lines, or you're frequently pulling in some tropical materials or other outside materials into your program, that array is going to consistently provide you with segregating and with informative SNPs within to genotype your program with. So each of them has their added benefits. If you'd like to go into a little more detail, please feel free to contact us and we'll talk with you about the benefits of each of the different aspects of these different types of genotypes. So next up is, how do you design a training set? So when you talk about designing a training set, for those of you who are much more familiar with this, you'll see that I've kept it pretty high level. There are ways to design a training set that are quite advanced. You can use optimization techniques. You can use clustering analysis. You can use a combination thereof. But a great place to start is to consider your historic data and your founder lines. So your founder lines, particularly if you have progeny directly off of those founder lines, so you have about 200 lines that you can get genotypes and historic data on, that can be a great base for your training set. That can be a great first training set for that first year or two of genomic selection. And the other thing you want to consider as you look at your historic data and your founder lines is to make sure that it represents your genetic diversity as it sits in your program right now. So essentially, when you look at that principal component analysis, you want to make sure that it's overlapping with that your training sets, so your historic and founder lines are overlapping with your current target sets. So essentially, those target sets are your current progeny. If your founder lines and your historic data aren't representative of that, then you need to consider how you can begin collecting that data so that you make sure you're representing the genetic diversity. And that just comes down to taking samples of those progeny as they are, trialing them, genotyping them, and beginning to construct a training set out of those. Along those same lines, you want to make sure you have phenotypic variation. Phenotypic variation often sits right there next to genetic variation, but it is important if you have a particular phenotype and you want to make sure that the target set and the training set, you can expect them to have similar phenotypic variation. A good example of this is when you have a training set that's from a different market segment or a different maturity than your target set. Often your phenotypes are going to differ pretty significantly. So it's important that you select a training set that's going to be representative of the materials that you're interested in predicting onto. And then finally, you can consider identifying clusters using market class, maturity, and other common classifications. So one of the, if you don't have the option of running that kind of clustering analysis in a formal way, you can often find that when you start running those cluster analysis, you can immediately start identifying the same things that you're already sort of clustering together in your breeding program in a natural way. So selecting training sets from within those clusters for the target materials that sit within those clusters. So essentially, just like I said before, with market class and maturity, those are great ways to help you identify training sets. The next thing, should I run genomic selection early or late in my program? I so wish there was an easy answer to this one, but really the answer is going to vary pretty significantly depending upon what type of crop you're working with. So for a great example of this is, I think a lot of the genomic selection literature you see out there is actually around grain crops, where you're selfing and you're having to genotype at each generation. You actually want to run genomic selection at. And so that means that you're going to have a different approach to genomic selection than you might in a clonal crop, where your genotypes remain static. And so essentially you can genotype really early and then continuously take advantage of those genotypes as you make selections and go through the process. So that's where it becomes difficult, and that's really where it comes down to an operational question. So what's your genotyping budget? Do you want to spend more on genotyping up front, but then you can get rid of those bad lines, those poorly performing lines earlier in your program? Or is your genotyping budget perhaps a little bit smaller? You might also consider, is your data quality not that great early in your program? If that's the case, you might also then again want to consider genotyping a little bit later. There's a couple of different ways to look at all of this. And again, this is where I think it would be great to have a discussion with you around the details and what would fit best within your program. So finally, in terms of frequently asked questions, just this idea of whether or not genomic selection is cost and time effective. So we've put together a fun tool that you can use to run some analysis around, given some basic information about your breeding program. So you're going to choose the type, self, clonal, or hybrid. For the hybrid, I'll just say you don't see it here, but we do include the test crosses as a consideration of that. Then you input some information about the number of crosses you want to start with, and that's going to educate the number of families you have moving forward. What's your cost per trial plot, your cost per nursery row, your genotyping cost? How many seasons are you going to run this program for? And then you can consider all of those things, as well as some options around, say, the number of selves and the proportion of lines you want to select if you want to get into some more details. And then you can see the outputs. So we're going to provide you with access to this after the webinar. And I really hope you'll go in and take a look and see some of the different options. In this case, what you see is a program that actually the web page is going to default to for you, where you can see I put together a program for, in this case, a self crop. And I was able with genomic selection to reduce the number of seasons. So I've gone from 9 to 12. So definitely time effective from that standpoint, but I've also managed to reduce the cost. And that's largely around this idea that I didn't have to trial nearly as many rows with this program as I did with the traditional breeding program, to come out at about the same, with about the same selection intensity. So I can expect to come out with about the same genetic gain overall, but for less money and overall much faster. So just to wrap it up today, I just want to give some quick tips for getting genomic selection to work within your program. So one of the things we like to recommend is that you start smart and assess as you go. So when I say start smart, I mean, think about how genomic selection is going to fit best within your program. Where does it fit operationally? How much, that gets into your genotyping budget, and figuring out, things, just the technical details of, like, how do you tissue sample, all of these kinds of things. So for you, it might mean to start slow. It might mean you start off in one particular program and you start tracking a few crosses with genomic selection. For somebody else, it might mean that you start genotyping a much wider swath of your material and you start running genomic selection at a particular stage of your program. Either way, it needs to fit within your pipeline and fit within your operations. The second thing, and you're probably starting to feel like I'm harping on this a little bit, but but it's just that important. The second thing is to organize your data really early. And so the sooner you can get a feel for your historic data and the data you're collecting right now, see how well that data is performing. Do you have standard protocols for all of your programs so that everybody's collecting, say, maturity notes in the same way? How are your yield measurements? Are they really representative of your materials? All of these things all of those kinds of questions you can answer when you start organizing that data and getting a feel for it. We highly recommend that you use cost-benefit analysis to work both genotyping and genomic selection into your budget. So that really gets back into number one there, and hope that you'll use the tool that we're providing as a means to kind of get a feel for what might be possible given your current genotyping costs. We'd also love to talk with you about your genotyping costs and ways that we might be able to work with you to sort of reduce those and make genomic selection a possibility within your wider program. Next, use the information you collect for genomic selection to track your program. This is one that really kind of gets back into the business side of things. When you genotype and when you sequence, you have a clear history of what exactly has gone on in your breeding program, and you have hard data to prove that. And so you definitely have a secondary business reason to begin using genomic selection besides the really great added benefits of time and cost effectiveness. It adds this extra layer of protecting your business and your program as you move along. And then finally, and I can't stress this enough, choose an analytic platform that keeps the breeder central to the process. And so what we've done as we put this presentation together is try to put this presentation together in such a way that it's very approachable for a breeder and a field breeder in particular. And the same is true of Breed, our analytics platform. And so we hope that you will discuss with us how you can actually run genomic selection in a user interface. You don't have to rely on R or Python in order to make that happen. And so you can get genomic selection incorporated into your program without losing that extra layer of breeder's knowledge that's so vital to your process. And with that, I just want to say thank you so much for your time. And I hope we have lots of questions, but if you have additional questions or you'd like to go into more detail, please feel free to contact us. We'd love to talk with you. Thank you very much.
Thanks so much, Kendra. Well, now I'd like to do a short Q&A session. If you have a question for Kendra that you haven't asked, please feel free to type it into the chat box now. Well, Kendra, the first question we had was, what exactly is a training set? We had this a little earlier on in the presentation, so you might have answered it, but feel free to answer it again.
Okay, so your training set is your set of lines that you've collected both phenotypes and genotypes on. And what happens with it is because you've got those phenotypes and genotypes together, you can use the algorithms that have been previously defined to actually identify patterns between the phenotypes and genotypes within that training set, and then use those patterns to predict onto your target set. That kind of brings into the idea of what is a target set, and a target set is that set of materials that you either have genotyped, or let's say you've only genotyped them, or they might be lines that are really early in your program, and so you have very limited phenotypes on them, or maybe you feel like your data quality is not that great early in your program, so you want to use predictions in addition. All of that goes into training versus target set. It also, when you talk about pattern identification, I do want to stress again, those patterns you're identifying in your training set need to be representative and work for your target set. And that's why we're talking about overlapping. So your training set is a set of materials that is phenotyped and genotyped and representative of the genetic diversity of your target set. All right, Alex.
Yeah, thank you so much. Our next question is, how do you prevent genomic selection from bringing along negatively correlated traits to yield, for example?
That's a great question. And I think that really comes down to a couple of different things. There are a couple of different things you have to do. You have to have a marker set that represents your entire genome. So just like anytime you make crosses, you're going to have a certain amount of linkage drag. So you need to have a marker set that's representative of the entire genome so that you can track what exactly is going on as you push things forward. You also need to be careful and think about how you're using genomic selection. The nice thing about genomic selection, though, is that because you're selecting out for the entire genome and the overall predictive performance of that line, and in general, you're going to run through multiple cycles of selection, it's not, you don't have to worry quite so much about linkage drag as you do with, say, a marker-assisted selection approach, where you have a very limited sort of window or view that you're taking when you look at marker-assisted selection. So overall, what I recommend is make sure your markers are representative of the entire genome, and then track the performance as you go along. If you track the performance and continue to use genomic selection, the overall probability of bringing along negative alleles is much lower, just like whenever you trial multiple times and make selections out of a population.
Super. Another question we got was, is there a minimum population size required for this type of analysis to be accurate? And can small breeding programs benefit from this?
Small breeding programs can absolutely benefit from this. This is where I'm going to get onto a little bit of shaky ground. So in general, the literature and just general experience, you recommend a training set size of about 200 lines. In reality, that's not always feasible. So when we talk to small breeding programs, the thing about small breeding programs is you often have a smaller pool of genetic diversity that you're pulling from. The end result of that is that you might want to use genomic selection just a little bit differently than the big players do. And so what you can do is you can begin by considering the genetic diversity within a given biparental cross. And instead of using, say, all of your historic data as your training set, we might consider using a portion of your biparental cross progeny. So you're going to trial your progeny, like say 75% of your progeny, then you genotype 100% of it, and then you use the results from that 75% to predict onto the results for the 25%. That progeny pool can then be smaller. And I don't want to give you an exact number because we really would need to talk about that and talk about what phenotypes you're considering, what your genetic diversity is. A lot of different things need to go into that. But you can get away with a smaller training set when you take that approach.
Thank you so much for that. Now, this question has a couple of different acronyms, hopefully you know. It was, can you comment about the accuracy of a genomic selection model, aka GBLUP, versus the accuracy of traditional ABLUP? According to literature, the GS model has always had less predictive power.
Okay, I'll be honest with you, off the top of my head, I would not feel comfortable commenting on the general accuracy of those different models, primarily because in my experience, those kinds of different models will perform very, very differently depending upon the data set. So that's going to get back into my previous answer of it's more important the data set that you're putting into something than it is necessarily of the approach or the algorithm you're taking. But if the person who asked that question, if you'd like to shoot me an email, and maybe we can get into a little bit more detail and hopefully I can understand your question a little bit better, I'd be happy to do that.
Okay, thank you. The next question hopefully is a little easier, and it's, does your software allow for prediction of three-way crosses?
So right now the software is set up to allow for prediction of two-way crosses. However, if you have F1s that you've genotyped, or if you want to produce, say, or you can pretty easily predict out the genotype of an expected F1 and then identify predictions off of that F1, push the F1 into the tool, and then use that F1 by cross, that would be a possibility. So it's not a direct way to make the prediction, but the tool can accommodate it.
Okay, thank you. And then what is the best way to manage sequencing and subsequent assembly and analysis data?
So in terms of sequencing, in general, what we recommend is that you work with a bioinformaticist to identify the best possible path for analysis of your sequence data. There are a number of different algorithms. Again, that's going to get into some details around the data set that you're using and what you want to accomplish with it. And so I think if you have a specific data set in mind and a specific question around it, be happy to have a conversation with you. But to provide a general, I don't think it would be fair to provide a general answer to that.
Okay, thank you. Um, and then let's see. How can we evaluate the accuracy of a selected model comparing with other models?
That's where you can consider the cross-validation scores in general. So if you're specifically looking at comparing your training sets and say how the model is performing for a given training set, which is usually your best way of assessing how you're doing with genomic selection, then you can take a look and directly compare the correlation scores for a given trait. So if your correlation score for a given trait is, say, 0.7 for one model versus 0.6 for another model, I would say that you have a better overall training set and model than for the 0.7 than the 0.6 one. Otherwise, I really, I don't know that I have a great answer for that other than to say, take a look at your cross-validation. And then another recommendation that I generally make is, especially if you're starting off with genomic selection, start off by running some experiments with genomic selection where you're also running essentially your own selections alongside alongside that genomic selection, and see how they compare and contrast. It will tell you a lot about the data you're collecting, and it will tell you a lot about how you're doing with genomic selection.
Okay, great. Someone else asked if you can use multi-trait genomic selection using this software.
At this point, no. At this point, we're set up for single trait genomic selection. However, the software will provide you with all outputs for all traits simultaneously, and so you can view all the traits alongside each other.
Okay, perfect. And then what approach would you recommend to start in a small commercial breeding program? GS in early generations or new hybrid combination prediction?
Okay, so if I had to choose between those two based upon where the technology is right now, I would choose genomic selection at an early stage. That's primarily because there are some great people working on this problem, and they're working on some great algorithms, but I've not seen an algorithm yet that does as well or is as far along for hybrid prediction as we are for genomic selection. We're actively working on this problem within Benson Hill and expect to have a solution. But for right now, in general, I would recommend you go with genomic selection, primarily because the technology is that much more advanced.
Perfect. And then I think we have time for one last question, which is, okay, so it says, you can predict the best crosses between heterotic groups. Could you predict what would be the best crosses within heterotic groups for population development?
Well, actually, the tool that I was talking about around which crosses to make is really around that development cross. So that's going to be around population development and around expectations of progeny. So when you can pull a distribution of progeny off of a cross between two inbred lines, obviously you're looking at some lines that have been inbred to a certain extent. In this case, it's to an F3 generation. And so you can understand the diversity and the expected performance off of those progeny from that development cross. In terms of hybrid crosses, in general, what I would recommend is that you take a look at some of the basic information that we started off with when we were talking about the development cross. So what's your genetic distance? What's your predicted, so your mid-parent value, things like that. There's other information you can pull out there, but again, I just haven't seen something that really strikes me as being an overall winner yet. Though again, I'm looking forward to having a discussion with you again here within the next year or so around Benson Hill's own hybrid prediction tool for that purpose.
Okay, well, thank you so much, Kendra. Well, I think that's all the time we have for today. So thank you so much, everyone, for participating, and I hope you found this information of value. I'd also like to quickly thank our technical assistants for helping us out today, Teresa and Kyle, who've been working behind the scenes to make this a successful event. Thank you again, and we hope you all have a terrific day. This is Alex Martin of Seed World signing off.